Good day, ladies and gentlemen. I am Alfli Bradilla Hameron. Together with my partner, Ms. Rina Bea Tagutungan, we will be discussing about gene therapy. Medical science has detected many human diseases related to defective genes. These types of diseases are not curable by traditional methods like taking readily available medicines. When we say people having defective genes, it means they're carrying diseases that are not curable by simply taking readily available medicines. Examples include severe combined immune deficiency, adenosine deaminase deficiency, hereditary blindness, fat metabolism disorder, blood disease, hemophilia, cancer, and many others. When we hear these diseases, we think of one thing. It's an inherited disease disorders, meaning to say, either the father or the mother's side have this kind of genetic disorder and is passed from generation to generation. What is gene and how important is it not to have defective genes? Genes is the basic unit of heredity passed from parent to child. They affect your development before you're even born and play a role in everything from your appearance to your personality. Genes contain information about your heritage and your risk for certain diseases. Gene therapy is a potential method to either treat or cure genetic related human illnesses. It's a medical approach that treats or prevents genetic related diseases. But how and in what way? In 1972, Theodore Friedman and Richard Roblin proposed that people with genetic disorders can be treated by replacing defective DNA with good DNA. Why do we need to replace defective DNA with good DNA? It is because genes that do not work properly can cause disease. Transplantation of normal genes into cells in place of missing or defective ones will serve as a drug to correct or treat genetic diseases. This is the gene augmentation therapy or replacement therapy. In the first picture, we see a cell with non-functioning gene. This non-functioning gene is the one that causes disease. Through adding DNA containing a functional version of the lost gene, or what we call replacement of genes that cause medical ill health, the method generally destroys the problem-causing genes. It helps the body to fight against diseases by adding genes to the human body. With that, the disease will be either prevented or cured because the cell is now functioning normally. But the question is, can the changes gene therapy causes be passed on to future generations? There are two types of gene therapy, namely somatic gene therapy and germline gene therapy. Somatic gene therapy is the placement of a human gene into a living person's somatic cells, cells that do not produce the eggs and sperm that in turn produce the next generation. Somatic cell gene therapy would aim to cure a disease only in the patient, not in the patient's descendants. Germline gene therapy is when DNA is transferred into the cells that reproduce cells, eggs, or sperms in the body. This type of therapy allows for the correction of disease. Delivery of therapeutic genes is strictly for the benefit of the recipient and not to be passed on to succeeding generations.
In 1985, Dr. W. French Anderson and Dr. Michael Blaise worked together to show that cells of patients with adenosine deaminase or ADA deficiency can be corrected in tissue culture. So, way back in 1985, si Dr. W. Anderson ag si Dr. Michael Blaise nag-work sila together or nag-collab sila para makita nila nga ang cells sa patient nga naay adenosine deaminase or ADA is ma-recovered pa in tissue culture. So, when we say adenosine deaminase or ADA deficiency, it is an inherited disorder. Inherited disorder is, for example, naay sakit iyang mama o cancer. And then, iyang anak is nagka-cancer pod. So, katunga sakit sa iyang anak, nakuha to niya sa iyang mama. So, mao na siya gitawag nga inherited disorder. So, ADA deficiency is an inherited disorder that damages the immune system and causes severe combined immune deficiency or SCID. And then people with SCID lack virtually all immune protection from bacteria, viruses, and fungi. And they are prone to repeated and persistent infections that can be very serious or life-threatening. And the main symptoms of ADA deficiency are pneumonia, chronic diarrhea, and widespread skin rashes. In 1990, the first approved gene therapy clinic research took place at the National Institute of Health, or NIH, under Dr. Anderson. So way back in 1990, ang pinakauna nga na-approve nga gene therapy clinical research is happened in National Institute of Health, or NIH, under Dr. Anderson. Now, is gene therapy safe? Well, the first gene therapy trial was run more than 30 years ago, and the earliest studies show that gene therapy could have a very serious health risk, such as toxicity and inflammation and cancer. Since then, researchers have studied the mechanism and developed improved techniques that are less likely to cause dangerous immune reaction or cancer. Because gene therapy techniques are relatively new, some risk may be unpredictable. However, medical researchers, institutions, and regulatory agencies are working to ensure that gene therapy research, clinical trials, and approved treatments are as safe as possible. Also, the National Institute of Health, or the NIH, also plays an important role in ensuring the safety of gene therapy research. And NIH provide guidelines for investigators and institutions such as universities and hospitals to follow when conducting clinical trials with gene therapy. Now, is gene therapy expensive? Well, according to my research, gene therapies are often unaffordable to those that need them the most. And $2 million a dose, only the very wealthiest can be cured with a single treatment. And depending on payers, this may not be accessible even in high-income countries. The first commercial gene therapy product, Gendicin, was approved in China in 2003 for the treatment of certain cancers. So way back in 2003, i-approve sa taga-China ang Gendicin, and kani siya ang pinakaunang commercial gene therapy product para sa treatment sa specific cancers, and it has been shown to be a safe and effective para sa treatment sa variety of cancers. And a number of cancers have been successfully treated using Gendicin. And then more than 10,000 patients have received treatment with Gendicin since it released to the Chinese market in April 2004. Due to some clinical successes since 2006, gene therapy gained greater attention for researchers. So, since 2006, ang gene therapy nag-gain daw nig more attention sa mga researchers due to some clinical achievements. In 2016, the Committee for Medical Products for Human Use, or CHMP, of the European Medicines Agency, or EMA, endorsed the gene therapy treatment called Strensilis that was approved by the European Commission in June 18. So, the Strensilis gene therapy treatment was approved by the European Commission in June 2018 and received a approval from the Committee for Medical Products for Human Use, CHMP, of the European Medicines Agency, or EMA, in 2016.
So, what is Strensilis? And what is it used for? So, Strensilis is a medicine used to treat severe combined immunodeficiency due to adenosine deaminase deficiency, or ADA-SCID. ADA-SCID is a rare inherited condition in which there is a change or mutation in the gene needed to make an enzyme called adenosine deaminase, or ADA.
Some studies transplanted genes to speed up the destruction of cancer cells. Gene or cell therapies have emerged as realistic prospects for the treatment of cancer and involve the delivery of genetic information to a tumor to facilitate the production of therapeutic proteins. So in several cases, genes were implanted to hasten the death of cancer cells with the delivery of genetic information to a cancer to stimulate the creation of therapeutic proteins. Gene or cell therapies have become viable options for the treatment of cancer. And this area of gene therapy still needs further studies before an efficient and safe gene therapy procedure is adopted. Now, what are the ethical issues surrounding gene therapy? So, gene therapy involves making changes to the body's basic building blocks, the DNA. It raises many unique ethical concerns. The ethical questions surrounding gene therapy and genome editing include: how can good and bad uses of these technologies be distinguished? And who decides which traits are normal and which constitutes a disability or disorder? And will the high cost of gene therapy make it available only to the wealthy? And could the widespread of use of gene therapy make society less accepting of people who are different? And last is, should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? To conclude, theoretically, gene therapy is the permanent solution for genetic diseases, but it has several complexities in its current stage, and it is not accessible to most people due to its huge cost, meaning gene therapy suit for those wealthiest people. Gene therapy have the potential to revolutionize the practice of medicine. Overcoming all its hurdles would change our lives forever.